virtual science pub. My name is Abby Putnam, and I am the early childhood educator at OMSI, and I will be your host tonight. Before we get started, I have a couple of things to tell you about what's going on at OMSI these days. First off, OMSI is open. Our current exhibit, Dinosaurs Revealed, is open through September 6th. There's animatronic dinosaurs, dozens of fossils, plus 2 full dinosaur skeletons. Advance tickets are highly recommended. You can also catch a show at the planetarium or theater and a tour of the submarine. Check out our website for the most up-to-date information and to view our COVID safety guidelines. I hope you enjoy tonight's pre-pub trivia and music by local Portland artist Stephanie Schneiderman. Putting on these shows takes a lot of work, and we have an amazing partner that helps make this happen. A big thanks and shout out to Celestream for providing the live streaming service for tonight's Science Pub. We really appreciate their support. Right. Join us for our 10th and last virtual OMSI After Dark, a summer beer fest. We'll be featuring new releases and flagship beers from Oregon breweries. The lineup includes Kolsch, Pale Ales, lagers, IPAs, and more. Tickets are on sale now. So visit omsi.edu/afterdark for more info. And we encourage you to put the pub back into the Science Pub experience by ordering some delicious food and tasty beverages from one of our partners from around the state. Today's event is set up just like our regular Science Pub programs. And for those of you who don't know what that looks like, we're going to begin with an alcohol-themed trivia game that is a warm-up for tonight's talk. So go ahead and grab a pen and paper so you can participate. After that, we'll learn about Prairie Voles, their social relationships, and how alcohol is involved. After the lecture, we will take your questions. You can submit them at any point during the event via the comments on Facebook or YouTube. We'll collect them and ask the speaker at the end. If you enjoyed tonight's lecture, please consider making a donation or purchasing a Science Pub pint glass. But don't worry, there's no pressure to donate. Our mission at OMSI is to inspire curiosity by creating engaging science learning experiences for people of all ages and backgrounds. So sit back, relax, and get ready for some fun. All right. So first off, we are going to be getting into our trivia. So this week, we have Annie Douglas, one of our amazing educators at OMSI, and my boss, joining us to play along live with you at home. So please give her a warm welcome.
Hey, thanks, Abby.
Hi, Annie. How are you?
I'm good. So excited to be here.
Oh, yeah. What are you most excited about to learn about during this lecture or about this lecture?
Well, I'm really excited to think about how Prairie Voles might be like us. They drink, they have relationships.
All right.
Sometimes that gets messy. I don't know.
I don't know. I haven't been in a menage a vol before, so yeah. We'll see what happens. All right, let's get started with our trivia game. So we encourage you to play along at home and compete against your housemates. You can play for bragging rights, or maybe who's going to do the dishes next. Annie, are you ready?
Ready.
All right, let's get to that first question. According to the 2019 National Survey on Drug Use and Health, how many Americans over 12 years old suffer from an alcohol use disorder? Is it A, 10.2 million people, B, 14.5 million people, C, 17.3 million people, or D, 25.1 million people?
Oh, those are also oddly specific. Also, over 12. I feel like if we're going down to 12 years old, that's a lot of age range to consider. So I'm going to go with the biggest number, D, 25.1 million people.
All right. Annie's going with D. Lock in your choices at home. The answer is 14.5 million people. So about 10 million people less than you thought, Annie.
Mm-hmm.
All right. Let's see. What's our next question? Question 2. According to the Diagnostic and Statistical Manual of Mental Disorders, 5th edition, how many points does an individual have to meet to be considered to have severe alcohol use disorder? Is it A, 0 to 1, B, 2 to 3, C, 4 to 5, or D, 6 or more?
I wonder how many points total there are. We don't know. Okay.
No idea.
0 to more. Wow. Okay. Well, I'm guessing it's not 0 to 1. That
Probably
seems
not.
too low.
Yeah.
I guess the most last time. So I'm feeling a little gun shy about D. So I'll go with 4 to 5. C.
Okay. Going with C.
Seems like you have to meet that many things to be considered to have severe. I don't know.
All right, we'll find out. It's no! It's
Man.
D, 6 or more, Annie. You got
Rats.
faked out by the first one. I'm sorry.
That's all right.
All right, let's find out what the next one is. All right. Heavy alcohol consumption is defined as blank drinks per week or more for men and 10 drinks per week or more for women. Is it A, 3 drinks per week, B, 7 drinks per week, C, 14 drinks per week, or D, 20 drinks per week?
Okay. Feeling a little more confident about this one.
Okay.
I know that men have usually higher body mass than women. So they probably have higher consumption to still be defined as heavy.
Okay.
And if 10 is women, I don't think it's double at 20. So I'm going to say C14.
C14, all right. You are correct.
Yes.
Yay. All right. Alcohol use disorder is the blank leading preventable cause of death in the United States. Is it the first leading preventable cause, the B, the third, C, the 5th leading preventable cause, or D, the 10th leading preventable cause?
Wow. That's tough. Preventable cause of death. I mean, I don't know if that's up there with like not wearing your seatbelt and like accident kind of stuff. Is that a preventable cause?
Sounds like it could be.
I think it
Yeah.
could be. Let's say it's B, the third. It's high, but not the highest.
Okay. Third. It is the third leading preventable cause of death in the United States. All right. Nice job.
Okay.
All right. Here's the tongue twister drugs.
Oh boy.
Which one of these drugs is not FDA approved to treat alcohol use disorder? Is it A, oxytocin, B, acamprosate, C, naltrexone, or D, disulfirin?
Okay. The only one I've actually heard of is oxytocin.
Okay.
And I feel like that's the feel-good hormone that you release when you like get hugs and eat chocolate and things like that. So I also don't know that you can administer that. I don't know if there's a...
Through chocolate?
Okay, yes.
Okay.
Just deliver it through chocolate.
All right.
So I'm going to say A, oxytocin.
Right. Final answer is A. And A is correct. Oxytocin is not FDA approved. Not chocolate approved. All right. In popular media, oxytocin is sometimes referred to the blank hormone. Is it A, the sad hormone, B, the angry hormone, C, the happy hormone, or D, the love hormone?
I think it's the love hormone. Chocolate and hugs, man. You gotta, yeah.
What if you don't love chocolate?
Oh, well, maybe it's the sad hormone. I don't know. It's D, it's love.
All right. It is the love hormone, oxytocin. All right. Nice job. All right. Prairie voles are socially, A, are they socially responsible? B, socially monogamous? C, socially awkward? Or D, socially distancing?
Well, I love all of these. I love the idea of a socially awkward prairie vole. But I'm going to guess, just based on the title of tonight's lecture, that we're going to talk about some monogamy. I think they're monogamous. B.
Right. Prairie voles might be socially monogamous. They're socially cute.
They are. Look at those whiskers.
And socially monogamous. Nice job, Annie.
Thanks.
All right. Approximately how many mammals display social monogamy? Is it A, 4%, B, 10%, C, 25%, or D, 50%?
Oh, that's so hard. Mammals. I don't think mammals are great at monogamy. So a 4%?
A 4% is your guess. And it's the answer.
Yes.
Nice. I probably would have guessed a little higher myself, but I think birds really are the ones who
Yep.
I'm thinking of, perhaps.
Yeah.
I don't know. I'll have to check those stats. All right. Where are prairie voles normally found, other than in these beautiful pictures?
Oh my goodness, that one's so cute.
I know. I love how they're always staring right at the camera. Are they found in A, Mexico, B, China, C, Madagascar, not the movie, or D, Canada?
I'm trying to think of places with prairies. I'm guessing they're found on prairies.
It's possible.
Which the only prairie that I know of is Canada. Those other places might have them, but I'm going to guess Canada because I am pretty confident there's prairies there.
Prairies in Canada. They are found in Canadian prairies. All right, nice. All right, this is the last question, so hopefully worth 3,000 points. You can win your trophy. When measuring pair bond strength, what is the main dependent variable in the partner preference test? Is it A, huddling, B, high fives, C, mating, or D, jumping?
The partner preference test. This is like the decathlon events of prairie vole relationships.
Sure.
Okay. Wow. I don't think it's high fives or jumping because that doesn't seem like you're hanging out with your partner. So huddling or mating. Again, I'm going to go with the title. We're talking drug, sex, and prairie voles. So I'm going to say C, mating.
C, mating. Oh, I guess, doesn't everyone love huddling together, though?
I do love a good huddle. Dang it, I missed the 3,000 points.
Sorry, Annie, your trophy will wait for next time.
Okay.
Yeah, but pretty good job.
Thanks.
Thanks for joining us, Annie. It was great to see you, and I hope you have some more fun learning about prairie vole, sex, and alcohol.
Awesome. Thanks, Abby.
See you at the museum. All right. Thanks, everyone, for joining us tonight, and I hope all of you at home had some fun learning something new. I sure did. So I would love to introduce our speaker for the night. Dr. Andre Walcott is a Black queer social neuroscientist who turned program manager who focuses on increasing diversity, equity, and inclusion in higher education. Andre started his research career as an undergraduate learning and memory researcher at Bowdoin College in 2008, where he received his BA in neuroscience in 2012. He then moved to New York City and explored the effects of resveratrol on the aging process at the Albert Einstein College of Medicine. He then decided that he wanted to move back to the West Coast and started graduate school at OHSU in the Department of Behavioral Neuroscience in 2014, where he received his PhD in 2019. Nice job. Andre knew that he didn't want to stay in a research career because he saw the systemic barriers that existed in higher education for individuals from historically excluded groups. This led him into moving towards a path in administration and eventually ending up in his current position, a program manager for 2 DEI enhancing programs at OHSU. Andre believes that representation matters and that everyone should be given not only a seat, but a voice at the table. It's my honor to introduce and welcome Dr. Andre Walcott. So thank you so much for being here, and take it away.
Well, thank you, Abby, for the introduction, and good job at Annie with all of the answers to those questions. They were pretty difficult, but you did really well. Thank you, everyone, for having me here tonight. I'm super excited to give this talk. So the talk I'm giving is kind of on my research I did in graduate school on Prairie Voles and understanding social relationships and alcohol's role in those in social relationships. So to start off, I like to give kind of a quick outline so we can all kind of be on the same page. Start with the introduction. We're going to go through some statistics on alcohol use disorder, what is a Prairie Vole, and kind of the relationship between the two of those, and then go into the main part of my studies that I did in graduate school. So my first study, I looked at alcohol's effects on established pair bonds, and then my next study, I looked at the effects of oxytocin on individual drinking in social settings. And then at the end, kind of wrap it all, tie it all up in a bow and send it off with you all, and then answer any questions you all have. So let's get started. So as some of these questions were actually some of these answers were in the questions. So in 2019, the National Survey of Drug Use and Health, they looked at how many people had an alcohol use disorder over the age of 12, and there was 14.5 million individuals in the US. Of those 14.5, 9 million were men and 5 million were women. And additionally, they saw that alcohol use disorder is the third preventable cause of death in the United States. That's behind poor diet and physical inactivity and tobacco smoking or smoking tobacco. Annie did bring up the fact of car accidents. I'm not sure if that was actually included in the survey. So that's something I also want to look into. As for the criteria, so how are you considered to have an alcohol use disorder? Again, we look at the Diagnostic and Statistical Manual of Mental Disorders, edition 5, and there's 11 criteria to be 11 points that you can kind of to be an alcohol use or have an alcohol use disorder. So some of those I like to point out are so important social, occupational, or recreational activities are given up or reduced because of alcohol use, increase in tolerance. Another one is withdrawal or taking alcohol to relieve withdrawal. So those are just a few of those, but so to be considered mild, have a mild alcohol use disorder, you have to hit 2 or 3 of those points. A moderate alcohol use disorder, 4 to 5. And then lastly, over 6 is having severe alcohol use disorder. So to understand the biological mechanisms that control alcohol use disorder, you have to do studies in preclinical models or animal models. And one animal model that has recently emerged as a good animal model to study alcohol use disorder is, of course, the Prairie Vole. So what is a Prairie Vole? So they're normally found in the grasslands in the central United States and in Canada, ranging from the Eastern Rockies, so over here in the west, and then to West Virginia in the east. and then going into the Canadian prairies in the north, and of course down here in some of the upper parts of the southern United States. They're highly, highly, highly social animals. They love to be in groups, be in pairs, just be around other animals and then kind of their offspring and their families. They're biparental and alloparental. So biparental means that the mother or the female and the male kind of parents will take care of the offspring together, which is something that's not very normal in most mammals. And an alloparental, meaning that the offspring will actually take care of other offspring. So kind of like older siblings will take care of younger siblings. And lastly, they're socially monogamous, but they're not sexually monogamous. So social monogamy is such an unusual thing, as we saw in those questions. Roughly about 4% of animals are socially monogamous, or mammals are socially monogamous. And so what is social monogamy? It displaying selective but not exclusive affiliation and copulation. They share nests with their partner, and additionally showing this biparental and alloparental characteristics really means you're kind of social monogamy or social monogamous. So as we were saying, about 3 to 5% of mammals are social monogamous. And so prairie voles are actually a really good model of human social relationships because of this. Additionally, you can actually study and understand how these long-term relationships actually form, or I will refer to them as pair bonds as their relationships. And 2 ways to actually study pair bonds is one, the partner preference test, which I will talk about more in the next slide. And then another one is the resident intruder test. And the resident intruder test, briefly, is when you have a male and a female in a cage together who are partners, and you take one of those partners out and you put in a stranger animal and you look at their aggressive behavior. So whenever an animal doesn't have a pair bond, they actually show less aggressive behavior compared to when an animal has a pair bond. They actually show a lot more aggressive behavior towards that stranger animal. And pair bonds and prairie voles are mediated by several neurotransmitter and receptor systems that are similar to those regulating actual social affiliations in humans. And some of those receptors and neurotransmitters are oxytocin, vasopressin, dopamine, the corticotropin releasing factor, or CRF, and the opioid receptors. So getting into the partner preference test, and I want to kind of drive this one home because this is kind of what most of this talk will be based off of, or the first section of this talk will be based off of. So I want to describe the partner preference test in detail. So the way the partner preference test works is it has these 3 different chambers. And within those chambers, you have we'll say this is a partner of a female partner. And then in this other chamber, you have a stranger female that the subject animal has never met. And the subject animal is actually, we'll say in this case, the male. And so these 2 side chambers, these animals are kind of tethered into these chambers. So they can't go into the other chambers at all. But you have this male is able to run around back and forth between the 3 chambers and deciding who they want to spend more time with. And as they said in the trivia, the measure that is used to understand the partner preference test is the huddling behavior. So they actually sit right next to each other and they sit in like motionless. And that is what you measure for how long they're actually spending together with their partner. And that's how you measure the strength of their pair bond or their relationship. So we know that prairie voles have this kind of social affiliation with their partners or with other animals. They're biparental, so they're very highly social animals. But then the other question is, do they drink alcohol? And of course they do, because if they didn't, I wouldn't be talking today, and none of this research would have ever been done. But I want to talk about some of the things that my lab, or the lab that I was in, and kind of stuff that they found out on prairie voles and alcohol drinking. So the first thing was they found that prairie voles actually have a natural preference for alcohol, and actually a higher preference for alcohol in groups. So natural preference when you give a Prairie Vole a bottle of water and a bottle of alcohol, let's say 10% of alcohol, and they're allowed to choose which one they want to go to, a majority of the animals will actually go towards the alcohol and drink the alcohol over the water. And one of the statistics that one of the grad students kind of calculated in the past, she said that Prairie Voles actually drink roughly 15 bottles of wine as like humans, but the difference is that Prairie Voles actually metabolize alcohol so much faster, so they don't get the drunk effects that we would get for 15 bottles of wine at any time. Next, alcohol consumption is actually influenced by drinking levels of peers. So when you have one Prairie Vole who's, okay, let's say a heavy drinker, and you have another Prairie Vole who's not really a heavy drinker, but a low drinker, and you put them in a cage, or they're in a cage together, and you give them a couple of days of drinking next to each other, they'll actually start to kind of come into a comparable amount of drinking. They'll still be different, but one will lower and sometimes one will higher their drinking. So kind of showing that there's somewhere in there, there's a social influence of alcohol drinking. Another thing we found out was that social partners actually help prevent relapse-like behaviors in Prairie Voles. So when you have Prairie Voles who are drinking, let's say for 20 days, and then they kind of go in this like quote unquote like relapse behavior. So you take the alcohol away, so they're going through relapse, and then you bring them, the relapse is when you give them alcohol again and they start drinking more. When you look at an animal that has a partner in the cage with them, their drinking actually is decreased. So again, there's something about this social aspect that is really kind of playing a role in alcohol drinking in Prairie Voles. And then lastly, something that I found out was that when a Prairie Vole goes through kind of withdrawal from alcohol, so if you take alcohol away from them over like a 24-hour period, they actually show an increased pain sensitivity. So it's kind of the equivalent of, let's say if someone is hungover and they always are wearing sunglasses in the sun because it's too bright. So that's kind of the same kind of thing. So there's an increase in kind of the sensitivity of their sensory system. So now that we know that Prairie Voles drink alcohol, they form these parabons, and they're kind of having the same receptor system, brain chemistry, kind of like humans in social affiliation. That's when I wanted to start getting into my studies. So we're going to start off with the first part, looking at the characterization of alcohol's effects on established pair bonds. So just some background on alcohol and human long-term relationships. So it has been shown that 73% of married men and 63% of married women drink alcohol. And for several decades, researchers have studied the relationship between alcohol consumption and marital status. Virtually every study has actually confirmed that there's a strong relationship between the two. In particular, research has confirmed that married people drink less than never married and divorced people. Additionally, alcoholics are less likely to get married than non-alcoholics. And if alcoholics do get married, they get married earlier, or they get often later in their young adult developmental cycle, the non-alcoholics. And lastly, alcoholics are likely to reduce use once they're married. So we call it the marriage effect. But also, they're more likely to get divorced than non-alcoholics. So one study, let me go back to this, is again, what we define as heavy alcohol consumption. So heavy alcohol consumption is defined as 14 drinks per week or more for men and 10 drinks per a week or more for women. And one study was looking at kind of what happens with heavy alcohol drinking in marriage and couples. So they saw that in 4% of married couples, both partners actually drink heavily, while in 79% of couples, neither partner is a heavy drinker. They also found that 12% of couples have only the husband as a heavy drinker, while 5% of couples have only the wife as a heavy drinker. And I do want to point out that this is definitely, a lot of these studies have been done in male and female identifying individuals, and not much has been done in kind of same-sex or same-sex partners. But some of that research has been done, but it hasn't been very extensively done. But with all of the previous study that actually explored substance use and divorce, there's been a pretty heavy limitation because all the research focuses on the substance use of actually only one member of the couple and not taking into account the other member's substance use. So, for example, they would ask let's say they'd ask the husband how much they're drinking, and that would be it, and then study how the relationship is going, but not actually look at both of them. And that actually changed recently in 2014. So in 2014, there was a study that came out by Leonard et al. And they looked at what happens when there's a discrepancy in drinking in long-term relationships. So when it came to divorce rates after 9 years of marriage, when one partner was actually a heavy drinker, there was a 50% divorce rate after 9 years of marriage compared to a 30% divorce rate after 9 years of marriage when neither partner was a heavy drinker. So that's kind of what we're thinking. Like, of course, alcohol abuse can actually split up couples and kind of destroy relationships. However, when they looked at when both partners were heavy drinkers, they saw that the divorce rate was very similar to when neither partner was a heavy drinker, and that was at 30%. So we put it as, yes, alcohol abuse can split up couples, but also it can also tie couples together in codependence. Within this study, they also took out some external factors. So they saw that socioeconomic status wasn't a factor. They saw that who they were drinking with wasn't a factor, and a couple more. But the one thing they didn't look at, because they're looking at humans, was kind of the biology and the biological mechanisms that happen in the brain. So from these studies, our lab was interested in understanding how alcohol actually plays a role in these bonds and in these long-term relationships or relationships in general. So we had to look at an animal model. And of course, being our lab, we looked at Prairie Voles and their drinking and social relationships. So in 2014, there was a graduate student, Alison Anaker, who's now a professor. She looked at kind of what happens in this period of Parabon Formation. So Parabon Formation is pretty much the first 24 hours of when a male Prairie Vole and a female Prairie Vole meet each other, and that's when they're forming this bond. So they're kind of formulating, and some of the brain chemistry starting to change. And she uses the partner preference test. So just to orient everyone to kind of what these graphs are looking at. So here on the y-axis, we're looking at the time spent huddling during that partner preference test. And on the x-axis, we're going to be looking at the kind of treatment. So either water or alcohol. And on these kind of open bars are the amount of time spent huddling with a partner. And then here, we're looking at the amount of time they spent huddling with the stranger. So in females, when there is actually access to alcohol or access to water, just water in their cage, when another male is also in there, and then they did their partner preference test, they saw that there was kind of a trend, we'll say a trend for significance between the amount of time they spent huddling with their partner compared to the amount of time spent huddling with a stranger. Interesting enough, though, when there was alcohol available to both the male and the female Prairie Vole in the same cage, and then the partner preference test was ran with the female being that subject animal and the males on the side, they saw that there was a very significant difference between the amount of time the females were huddling with their partner compared to the amount of time they spent huddling with a stranger. Thus kind of showing potentially that alcohol is causing a kind of an increase in the strength of these pair bonds in these female Prairie Voles. Of course, we have to look at males too. So when you're looking at male Prairie Voles, looking at their water, when only water was available to the male and the female Prairie Vole, and the male was a subject animal and the females was the partner and the stranger, you see that males tend to spend a significant amount of time with their partner compared to the amount of time spent huddling with their stranger. Interesting enough, though, when you start putting alcohol, when alcohol becomes involved, you see no difference in the amount of time that the males start huddling or spending time with their partner compared to with their stranger. Thus, alcohol is actually kind of strengthening these bonds in female Prairie Voles, but then in males, it's kind of destroying and deteriorating these bonds. And this is actually only over this 24-hour period. So when we talk about Parabon formation in Allison's study, the example which that she was kind of modeling was when a person has their first drink during their first encounter with an opposite sex individual. Of course, as we all know, this is not typical of alcohol drinking in humans. So what I wanted to do was try to make it a little more typical in the sense of let's look at the long-term bonds and the long-term relationships and how does alcohol affect those. So as I was saying, Allison was looking at the Parabon formation of kind of Prairie Voles, and my interest was looking at the Parabon maintenance. So explaining these two, because these are actually two very distinct behaviorally and biologically. So the formation of a Parabon, these animals will spend, of course, the time when the Parabon's formed, they spend more time with their partner, usually, than a stranger. And that's the huddling behavior. But when it comes to the maintenance of a Parabon, these animals will actually start showing, they'll spend more time with their partner, but also they tend to show more aggressive behaviors towards that stranger. So that's why that, like I said earlier, that resident intruder test is usually used during this maintenance period. Additionally, not only just behaviorally they're different, but also biologically they're different. Different receptor systems play a role in them. So for formation, there's this thing called the mu opioid receptor that's activated. And then in maintenance, there's the kappa opioid receptor during activation. So let's get into this first study. These are just the quick methods, but I wanted to bring these up because of looking at this. I'm sorry, this is supposed to be a female group over here, but looking at the way I ran this study. So I had Prairie Voles in a cage together, and then eventually they went to the same cage, but I put a mesh divider in the middle so I can actually figure out who's drinking what and how much each of them are drinking. So in male Prairie Vole groups and female Prairie Vole groups, we looked at when both the male and the female had access to alcohol. And then another treatment group was when only the male had access to alcohol. And the other one was only when a female had access to alcohol. And then lastly, when both male and female both had access just to water. Then I ran the partner preference test. And of course, being a neuroscientist, I looked at the brain mechanisms that were existing. Here's just a quick little timeline so you can kind of understand how this is ran. The first 7 days here was when you put the animals into the cage together. So they're kind of introduced to each other. They're able to mate. There's nothing happening. They're just in the cage cohabitating and living together. And then on day 8, that's when you start giving the treatment or the alcohol on board, or so they have access to the alcohol. And then on day 15, we had the partner preference test. And I wanted to show the cages because this will actually help you understand what this kind of look like. So these are 2 Prairie Voles in the cage, and there's this mesh divider in the middle. And within these bottles, so these ones without tape is with water, and the ones with tape are when they're actually drinking out, or they have alcohol in those. And so as you can see, you can really determine how much each of them are drinking because they're in the cage separately. But these mesh dividers allow for kind of some sensory systems. So they're able to see each other, they're able to smell each other, they're able to give some they're able to give some touch. So they do sit next to each other. But the biggest thing for these mesh divided cages was to figure out who was drinking what, and it ended to, and they weren't allowed to mate during this period. So looking over those 7 days of alcohol consumption, so just to orient everyone to these graphs, here we're looking at, these are in males, and then below here, these will be females. On this side, on the left-hand side, we're looking at alcohol consumption, and on the right-hand side, we're looking at alcohol preference. And preference of alcohol is actually calculating, dividing the amount of alcohol by the total amount of fluid consumed. So that's alcohol and water. And for A, we're looking on this left side, we're looking at alcohol consumed in grams per kilogram, and then days. And over the actual 7 days, when both the male and the female were drinking alcohol, the male tended to drink more alcohol overall, compared to when just the male was drinking alcohol and the female wasn't. But we did see no difference in the alcohol preference ratio, kind of meaning that it wasn't just alcohol that was affected, but also water drinking was affected. Then when you look at females, you kind of see the same thing, but it's just these last 2 days, there's a difference in alcohol drinking. And that is the female, that when the female was drinking more alcohol, when the male was drinking alcohol with them, compared to when the female was drinking alcohol and the male wasn't. But you do see that there is no difference in the alcohol preference ratio. So then looking at the partner preference test, we did females, of course, and then males. And these graphs are exactly the same as the last graph that I showed with the Parabon Formation. So the y-axis, we're looking at time spent huddling. X-axis is these treatments. And both ethanol is when both male and female are drinking. Of course, female only drinking ethanol, and then both drinking water. And black bars are the time spent huddling with a partner, and gray bars will be time spent huddling with a stranger. And you can see here that females spend a significant amount of time huddling with their partner compared to their stranger, which is pretty much almost zero in all three of these groups. So females weren't really affected by if their partner was drinking or not. So that wasn't an issue for them. But then when you look at males, you see that they do spend more time with their partners over their stranger, but when only the male is actually drinking, he tends to spend less time with his partner compared to the amount of time with the other groups. So this male, so there's something going on with not just the alcohol drinking is playing this role, but it's also what is the partner doing as well. So there's some social aspect that's happening that's really changing their behavior, and it's not just the alcohol drinking. So being a neuroscientist, you go through the behavior, and then you want to see kind of what brain regions are actually involved. So this is just a schematic of a rodent brain or a prairie vole brain or mouse brain, and looking at the different overlapping regions that are involved in pair bonding. So these regions that are in blue are specific to regions that have been shown to play a role just in pair bonding. The regions in red have been shown specifically to play a role in alcohol. And then these ones in purple are kind of overlapping regions for pair bonding and alcohol drinking. And so I looked at 18 different brain regions, and there were just a couple of them that actually showed some striking results. So one of the regions is the centrally projecting Edinger-Westphal nucleus, or we'll say it's the EWCP. So this is the location of the EWCP in a rodent. And then this is a small, tiny little region of the EWCP actually in a human. And so the EWCP has been shown to be sensitive to alcohol drinking and consists of different chemicals that contain neurons that project to several other different regions that play roles in addiction and also in stress. So when I looked at the prairie vole drinking and I wanted to look at this EWCP region and the activation in this region, I looked at all three of those groups. And so here on the y-axis, it's the number of cells that are activated in this region. And I saw that when any time the male was actually drinking alcohol, that there was always an increase in activation of this region compared to when they were just drinking water. And this was independent of if their partner was drinking alcohol or not. And that's the same exact result we see in females too. We see that it's independent of their partner's alcohol drinking, but if they are drinking alcohol, then they show higher activation compared to when animals who are just drinking water. The next region looked at was the Perioctal Gray, and it's like this tiny region here also in humans. So the Perioctal Gray is involved in a bunch of different things. So it's involved in the perception of pain, it's involved in fear and anxiety, it's involved in the production of sound, and also it has been shown to be involved in social attachment, it's been shown in maternal attachment and also kind of romantic attachment in humans. So we thought this would be a great region to kind of look at for this study. So here is an actual brain slice of a prairie vole. And this is the Perioctal Gray region. So I outlined it. And there's 3 different regions in there. Here's the DM, or the dorsal medial. Then we have the DL, which is the dorsal lateral. And then this region here is the lateral. And we were looking at activation of this region after all of these behavioral tests in alcohol drinking. So and this will only be in male prairie voles, so I saw no difference in female prairie voles. So in male prairie voles, when I looked at the total number of cells in the region that lit up, the animals that only had alcohol when the male group and the female didn't showed an increase in activation of this brain region compared to when they were both drinking alcohol or both of them were drinking water. So of course, it's such a big region. You want to break it down a little bit to see if there's something specific going on. There was nothing happening in the dorsal medial region. There was nothing happening in the dorsal lateral region. And of course, there had been some region that was causing this difference, and it's actually this lateral region. So here, these regions here are playing this role. So there's higher activation. So I also want to point to this again, because I think this is the point I really want to drive home, is the behavioral studies, the partner preference test, when only the male was drinking, the female wasn't, you saw a decrease in the amount of huddling behavior in those animals. But when you look at the Perioctal Gray, you see an increase in activation of this region. So we are kind of hypothesizing that this region, this Perioctal Gray region, might be playing a role in causing this decrease in partner preference or pair bond behavior. And so in the last couple of regions I looked at was the periventricular nucleus and the supraoptic nucleus. The reason why is because they both produce oxytocin and vasopressin. So as many people know or have heard, that oxytocin is called this love hormone. So oxytocin and vasopressin, they're actually very similar chemicals. There's only 2 differences in their chemical chains. So here and here. And they both have been shown to play roles in Parabon Formation. They've been shown to play roles in sexual behaviors and reproduction, female lactation, emotional regulation, pain and stress, and also rewarding behaviors for food, drugs, and sex. So looking at this periventricular nucleus and the supraoptic nucleus, we wanted to look at oxytocin, the number of oxytocin cells, and then also the number of vasopressin cells. And so these are 3 images from brain regions in male prairie voles. This one is when both of them both male and female drinking alcohol. This one is when only the male is drinking alcohol. And this one is when both male and the female drink water. So here in this graph, we see that independent of kind of what their partner's drinking, when the male is drinking alcohol, he tends to have decreased number of oxytocin cells in the periventricular nucleus. But then I also looked at vasopressin, and I saw no difference in those regions. And of course, if you do it in males, you should 100% also do it in females and see what goes on. And in female prairie voles, I see the same exact thing. So this oxytocin is potentially not playing a strong role in pair bond maintenance, but however, it does play a role in alcohol drinking. So we see that there's a decreased number of cells in both male and female prairie voles when it comes to alcohol drinking. So just a kind of a quick summary of what I talked about, because there's kind of a lot right there. So we saw that alcohol consumption is actually affected by the drinking status of their opposite sex partner. And then we saw that when there's a difference in alcohol drinking between partners, it leads to males spending less time with their partner. Compared to females, there was no difference. And with that behavior, the alcohol drinking in the males, we saw in the male-only group, we saw there was an increase of activation in the Perioctal Gray in these male Prairie Voles. And lastly, alcohol consumption leads to an increase of activation in the EWCP, or the Edinger-Westphal nucleus, and a decrease of the number of oxytocin cells in the periventricular nucleus. That's all independent of the drinking status of their partner. So from those studies and finding out that oxytocin kind of potentially played a role, led me into my next group of studies, and I wanted to explore oxytocin's effect on kind of individual drinking status in social settings. So the FDA has a couple approved pharmacotherapies for alcohol use disorder. The first one is Disulfirin, and this was approved in 1951. And what happens with Disulfirin is when it's taken with alcohol, it actually causes the adverse effects. So pretty much when you take it, you go through withdrawals while you're drinking alcohol. So it kind of forces people not to drink because they don't want to go through those withdrawal facts or go through those withdrawals while they're drinking. The next one that was approved was Naltrexone, and this was approved in 1994. And this one, it actually decreases the desire for alcohol drinking just overall. And then we have a Camprosate. This one's a little more complex. So this came out in 2004. It's not fully understood the mechanism, but there is some potential ideas of what it does in the brain, but it's also decreased the desire for alcohol drinking. So these three pharmacotherapies have not been very successful in treating alcohol use disorder in all patients. And they're only modestly better than kind of the placebo or like saline or anything that's not an actual like pharmacotherapy. So really, to discover new pharmacotherapies, people have to use kind of the preclinical animal models before you can give them to humans. So one animal, of course, that we're definitely going to talk about here is the Prairie Vole. And the other thing is, what about oxytocin? So we saw something in my last studies that oxytocin played some type of role, or oxytocin is affected in alcohol drinking. So there's been a couple studies that have looked at, in humans, oxytocin and alcohol drinking in humans. So the way that oxytocin is usually given in a human is actually, it's actually sprayed into your nose, which is interesting because most people think it's like an injection. So you can do the injection in animals, but you also can kind of spray it in their nose as well. But these studies in humans, they were all sprayed with oxytocin into their nasal cavities. So they've seen that oxytocin actually blocks alcohol withdrawal in humans. They've also seen that oxytocin decreases kind of the cue-induced alcohol craving. So cue-induced being, say you walk into a room and you see a label of, let's say, Bud Light, and that makes you kind of like, ooh, I'm craving alcohol now. And so you have an increased alcohol craving. So oxytocin actually does decrease that alcohol craving. So say that person walked into a room, saw that beer label, and wasn't craving it as much as someone who got like a placebo or a fake drug. Another study, again, was decreased neural reactivity to alcohol-related cues. So this now takes it a step further and looked at some brain scans and looking at that some of these regions that are dependent in rats and in humans, they saw an increased oxytocin receptor expression when there was a decrease this neural activity. And lastly, there was one study that actually looked at when you give oxytocin over a period of time or across time, they saw that there's a decreased alcohol consumption in a cancer patient. This was actually, to my knowledge, the first study that actually saw oxytocin treatment over time decreasing alcohol consumption. However, I do want to point out that this study was only done in one patient. So of course you have to do more you have to have larger sample size and a large amount of individuals in a study to actually understand if it actually works. So we know this oxytocin stuff kind of potentially and has some pretty promising results in humans. But of course, there were studies that had been done in rodents to figure out kind of what are the brain mechanisms and how does it work in these animals. So it's been done in mice. They show the oxytocin actually decrease the alcohol consumption in a paradigm called the 2-bottle choice. And that's kind of what we saw in my in my study. We looked at water drinking and alcohol drinking in 2 different bottles. And so they saw that there's a decrease in just the alcohol drinking in mice. In rats, this study looked at alcohol preference for a 5% ethanol vodka cruiser, which I didn't know what a vodka cruiser was when I read this. And I looked it up, and it's a type of alcoholic drink that is, I guess, from New Zealand. So they saw that there's a decrease in the alcohol drinking of this vodka cruiser in rats. And lastly, there was one study that actually did oxytocin drinking in Prairie Voles. And this was in 2017. And they saw that over time, if you gave oxytocin to daily injections of oxytocin to Prairie Voles over, let's say, 6 weeks, I believe, they saw a decrease in this alcohol consumption. So there's some pretty promising results, even in rodents and not just humans, that oxytocin actually decreases this consumption of alcohol. However, I do want to point out that all of these studies were actually done in isolation. So isolation being there was one animal in a cage, and they inject the animal, let's say, with oxytocin, and saw what they did with alcohol consumption and alcohol drinking. That's great, but the problem is that's not how humans kind of work. Humans, of course, we're social. We're very social animals. So we are trying to figure out how can we mirror this into kind of in human behavior. So there should be something done in kind of a social setting. So how do people actually study alcohol drinking, social alcohol drinking in rodents? Because again, we all know that alcohol is actually influenced by different social settings in humans. And when we do study alcohol consumption and the effects of pharmacotherapies, it is, again, usually done in a single housed animal or in a semi-social housing cages. So the first way that a lot of people do it is you have 3 animals in a cage, and there's let's say 2 bottles or 4 bottles or 2 bottles of water and 2 bottles of alcohol or something. And you see how much is consumed within that cage, and then you divide it by the number of animals. Of course, that's not very sensitive to what happens if animals drink differently. So you don't know on an individual level if things are changing and if there's differences in this alcohol drinking. Another way is this mesh divided paradigm, which I did in my last study, and looking at how each animal is drinking in this semi-social environment. But of course, these animals aren't actually allowed to have a full social environment. We're not doing studies in humans where we have a divider between them and looking at their alcohol consumption and then giving them, let's say, oxytocin injection. Then we have these last 2, which are definitely newer, but people have definitely done these in the past. Video tracking of social animals, and also using a radio frequency tracking system in animals. And so here, you have all the animals in the cage, and you actually have to watch all these videos and see how much time this specific animals by each bottle. And that's how much you divide, and you figure out the ratio of how much you're actually drinking. And that's the same thing here. When you have a RFID or radio frequency identification tracking system, you see how much each animal's by the bottle, and then you create the ratio. And the issue, of course, with these 2 is how inaccurate they are at measuring alcohol consumption still at an individual level, because you're kind of guessing that the animal is taking a certain amount of drinks during that period of time, because they could be taking different sizes of drinks when they are by the bottles. So the cool thing that our lab actually found was this company that created these cages that animals can live in the cage together, like socially housed, and then go into these kind of, like, let's say we'll call them tunnels. And when they go into a tunnel, there's an infrared beam. So we know that the animal's in there. And then they go further into the tunnel, and there's another infrared beam. And we know that the animal's actually close to the bottle. Then we use RFID technology. So I inject the animals with a kind of a tracker, and then there's a reader that tracks each animal, that knows which animal is actually in the cage. And then these bottles are actually on like a scale. And so whenever the fluid is coming out of the bottle, the scale, of course, will change. And we know how much which specific animal is actually drinking. And so this is probably one of the only systems that we know of that is like this high tech, and you get so much information out of it, and understanding on an individual level how much each animal is actually drinking. So for my next studies, here's just a quick timeline. So I gave those injections of those RFIDs, and they had 2 to 3 days to recover. Then I gave them another couple days to get used to being in those cages, because they are new cages that they don't live in all the time. Then they had 5 days of just water in both of those 2 bottles, because not only is there a bottle here, but there's also a bottle back there. So there's 2 different bottles. And then one of those bottles, I would put 5% alcohol into it, and another one was just water. And then I would inject them with oxytocin, inject them with kind of a placebo, and then inject them with oxytocin. So for what made this study even more interesting, because when I was thinking about this was, okay, in humans, let's say someone is hungover and they are trying to they have a headache the next day, and they're trying to get some ibuprofen. So you take the ibuprofen by yourself, but you're not giving it to, let's say, your partner as well, just because they're around. So I had 2 different experiments here, and I looked at what happens if one entire cage all of them get oxytocin, or all of them get saline. But then we have the mixed experiment being, what happens if half of them get oxytocin and the other half get saline? So will there be a difference between the alcohol drinking between these 2 animals in these cages? So specifically, again, we were looking at alcohol drinking, and I looked at hours 1, hours 2, or hours 3 after injections, hours 6, I think 12 and 24 hours after their treatment of oxytocin with these animals. And we're looking at here in this graph, the amount of alcohol they consumed. And here, this is again, the alcohol preference ratio we looked at earlier. And we saw that on that treatment day, which is day 8, you can see that there's a significant decrease in the amount of alcohol consumed in these animals. And these are specifically, again, the mixed cages. I want to be blatant about this. So I have the across data, but I didn't show it here. But in the mixed cages, you see that these oxytocin treated animals in these cages show a decrease, while those animals that received the placebo or saline didn't show any decrease. So thus showing that oxytocin does affect their consumption of kind of fluid. But again, we saw that there was no difference in their preference ratio. So it's not just their alcohol that's going down, but also their water drinking is going down. And I will say that even though we saw water drinking going down and alcohol going down, there was no health or behavioral issues in these animals. There was no difference. So they were all healthy. So we're not quite sure what it could have been, but their drinking did go down. And then after the first 6 hours, that's the same thing. We see on that first day that there's a significant decrease in the alcohol consumed in these animals. And again, I said I looked at 12 and 24 hours, but I didn't expect to see anything. I didn't see anything because oxytocin has a pretty short half-life, so it kind of leaves the system pretty quickly. So this is kind of the preliminary data of looking at maybe oxytocin is a good drug to actually use to look at alcohol consumption, maybe decreasing alcohol consumption. And of course, being a neuroscientist, you have to look at the brain regions. So I looked at a couple of brain regions and seeing if they correlated with alcohol drinking and the activity in those regions. So first, of course, I looked at the EWCP because that's one of, I would say that's one of the lab's favorite, it's definitely the lab's favorite brain region. But we looked at kind of correlation. So seeing if the activation in the brain in this EWCP region was correlated to alcohol consumption. And there was a correlation between the two when this was looking at the first hour of alcohol drinking. Then when you look at 24 hours, you see the same thing, that there is a correlation between the two. So this brain region is pretty closely, there's a big correlation with alcohol drinking. And then lastly, looking at BEC's blood ethanol concentrations in these animals, and you see the same thing, that the blood ethanol concentrations correlate with the activation in this brain region. So start to sum of this all up for this specific experiment. So we saw that peripheral oxytocin treatment decreases alcohol consumption and water consumption under social settings. That again doesn't decrease the alcohol preference. We saw that there was activation in the EWCP brain region, and it relates to the alcohol consumption in the first hour of drinking, and then after 24 hours of drinking, and the blood ethanol concentration. And this is actually the first study to find the effects of a potential pharmacotherapy for alcoholism in a mixed treatment social setting. So there hasn't been any studies that actually did this, looking at how in the social setting, how animals housed together and giving them different treatments. So the overall kind of conclusions of kind of all of these studies together is that discordant alcohol drinking between partners has kind of a sex-dependent effects in Prairie Voles, and potentially the Perioctal Gray might play a region in these. We know that alcohol disrupts the oxytocin system in the brain, because in that first study, we saw there was a decrease in the number of oxytocin cells in the brain after alcohol drinking, that a treatment of oxytocin leads to a decrease in water and alcohol consumption, and that the biggest thing is that when people are studying drug treatments in rodents, they have to take into account the sociality of these animals. These animals are social, we're social humans, so of course you have to kind of try to mirror or model that in the rodents. So as for some potential future directions that someone potentially might take down the line, maybe understanding how that Perioctal Gray plays a role in the relationship between this discordant drinking and the established parabons. Try to figure out why there's these sex-dependent effects. So why does it only affect males in these long-term studies, but not females? And maybe there's something else going on. And then lastly, really determining the mechanism that leads to a decrease in alcohol consumption after this oxytocin treatment in these social settings. I think this is, of course, very key and a big one. And that's kind of for myself, it's like the biggest takeaway from all the research I did is like really understanding how this oxytocin treatment does affect animals in these social settings. And so with that, I want to thank you all for being here tonight. I love giving these talks. I love talking about my research. It's always been so much fun. So I would take any questions if anyone has any, and thank you all.
Wow, that was fascinating. Thank you, Dr. Walcott. A few questions. My first question is, you talked about like the behaviors when they were meeting stranger mates. What does an aggressive prairie vole look like?
Yeah, so aggressive prairie voles, if you think of, it's funny, I have two cats and I think of my cats because whenever a cat kind of jumps up, they jump up at you and they kind of like stand on their hind legs. That's kind of what a prairie vole does too. And so they show this kind of like, like I'm a bigger individual than you and trying to be aggressive. Sometimes they will kind of lunge at them. So those are kind of some behaviors that you see.
Okay, cool. And then my second question is, who buys the Prairie Voles alcohol in the wild?
So that's a good question. No one is out there like, here's some wine from the Prairie Voles or anything. But we have seen in animals that they will actually ferment some of the, like wait for wine or grapes to kind of ferment, and they try to wait for those things and then they get drunk off of it. So it's kind of sometimes naturally these will happen. Prairie Voles, I don't know if they do that. I don't think we have any studies on that, but Prairie Voles do. You'll notice a Prairie Vole because the way the Prairie Vole runs, it kind of waddles their butt. And so their butts are waddling. And so it's so much different than a mouse. And Prairie Voles, there's so many, not Prairie Voles, but specifically just voles. There's so many voles in Oregon. And so whenever your garden is getting eaten up, it's probably a vole.
No, there's something eating my stuff. So yeah, I've heard about like different animals getting intoxicated. Like a lot of birds will also get intoxicated from eating like overripe berries. Did your study, like, I guess you can't ask an animal's opinion, but do you think that like animals like being intoxicated, or is it like just like a feeling? Yeah, I don't know. I honestly, I don't know. I don't know if we can say like, because we don't want to put too much like human behavior on it, but they do prefer, majority will say, if you, let's say, take 30 animals and out of the 30, majority of them will actually want to consume alcohol over water. We'll see it. Okay.
Yeah.
Interesting. Okay. Now a slightly more serious question. So some people, I think that there's studies that have noticed that people have started drinking more during the pandemic recently. Do you think that this is a trend that will continue, or do you think people will go back to drinking less? I know that's not necessarily your.
Yeah, but that's a very good question. I personally, I think, I mean, I'm definitely not a complete expert on everything, that's for sure, of all alcohol drinking. But from my own opinion, I think people will start to drink a little less because they're not going to be in their homes. But the other flip side is potentially some people might associate their homes now as like a bar-esque, like, oh, I drink so much more at home. So they might drink more when they're at home, but less kind of socially.
Yeah, interesting. Yeah, I was thinking about you were talking about social monogamy versus, I guess, reproductive or sexual monogamy. And are the voles socially monogamous from one mating season to another, or is it just for mating season? Because you also talked about their offspring. It was not allopatric.
Alloparental.
Like raising. So that makes it sound like they must be over multiple seasons.
Yeah. So voles. So voles tend to kind of stay where they're kind of like raised in a sense. So prairie voles, they will. They will. Let's say they copulation will happen and mating will happen, and then let's say 21 days, 21 to 23 days later, they have their offspring. But then usually they'll get pregnant like right after that. So it's like, it's quick. So they can have, it's definitely higher in some seasons.
Sure.
But they will keep mating. They'll mate with other voles, but I always like to put it as, they'll mate with others, but they'll always come back.
Nice. Do they live in, I don't know if you mentioned this, do they live in like social network, like underground tunnel networks? Because I know like prairie dogs do or something like that.
So they do live in, they're not technically, I wouldn't say they're not, they do burrow, so they definitely do go underground, but they do live in kind of a social like setting together.
Yeah. Awesome. Cool. All right. Enough of some of my questions. Someone asked, do males and females have the same amount of oxytocin naturally occurring in their bodies without outside influences such as
That's a good question. Yes, from what I can remember, there was no big difference between the amount of oxytocin in either of those, looking at males and females.
Okay, cool. Are there natural ways to increase your own oxytocin levels?
Yeah, there is. You go hug someone, being around people. I mean, that's why they call it kind of the love hormone. It definitely, your oxytocin will increase, definitely in like a social setting.
I'm just thinking about like the physical nature of a hug, and like if it has to do with that hugging proximity situation, or if there's like actual like a physical, like physical kind of response or not.
Yeah, I don't know. I mean, I would say probably. Your mood can definitely change. I mean, if you hug someone, you're like, oh, you're like, this feels like, sometimes you're feeling sad. You're like, I just needed a hug.
Yeah, totally. Another question is, why do you think we don't have more studies and research on how to fix alcohol abuse or how to help people with the alcohol abuse?
Yeah, I think when it comes to alcohol, we kind of deem it as, we call it kind of like a messy drug, because you don't know exactly where it's working. It can work anywhere. I mean, there's studies that are done in like, let's say cocaine or like methamphetamine, and you kind of know what receptors in the brain kind of are for sure. And we know that it's from the alcohol, but alcohol kind of goes everywhere. It like, so it's like it's kind of like a dirty or messy drug because it's just a widespread. Mm-hmm. So I think there definitely is a lot of studies on alcohol. I mean, there's conferences that I would go to in grad school that it's only on alcohol addiction. But I think it's less, you hear less about them because alcohol is such a social, it's such a socialized thing. Like a lot of things in Portland, if you think about it, they're like, hey, let's go bowl and drink. Let's go to an arcade and drink, or let's go watch a movie and drink. I mean, that's everywhere. So it's a little more acceptable, I would say, is like people will just go start drinking and it's acceptable. So I think there's less research. I mean, there's a lot of research on it, but you don't hear about them that much.
Did you look at any studies that looked at like alcohol? I forget the technical term for alcohol dependency. Sorry. Did you look at it like beyond the United States at all? I know these are like Canadian, North American bulls. So
yeah, I did not. I definitely did not. I did not for sure.
Yeah. It's interesting because it is such a social drug. It's like regulated and long-standing and part of so many traditions and things like that it seems like almost taboo to attack sometimes. Or
yeah, and it's true. And then it's always like when people, I mean, I've definitely heard people be like, if someone comes to Portland or something and someone's like, oh, I don't drink, and you're just like, oh, so what do I do with you? Like, I don't know what to do. And you're like, oh man, it's so socialized and it's like ingrained in everything that we do that it's true. It's like when it's you get kind of taken aback sometimes when someone's like, oh, I'm drinking. You're like, oh, I'm like, oh, that's great. Like, yes.
Yeah, I know. Have you changed any of your alcohol consumption habits after studying voles? Or that's personal, but you don't.
No, yeah. I mean, I definitely would say it is just seeing kind of what it does in general. I would say for sure.
Yeah. You said that the voles also had withdrawal effects. Like they had sensitivity to pain. Did they have sensitivity to other things? Like, did you put little sunglasses on them and shine lights at them, or like make loud noises or anything like that? Like,
I wish I had little vole sunglasses. That would be amazing. If someone wants to make some of those, let me know. That'd be great. And I just want a little pair. So yeah, someone Etsy, you'll have some buyers. That study only looked at kind of pain sensitivity, and pain sensitivity. The way that study kind of worked was you kind of do like a little poke on their paw, and it's when they move their paw, that's kind of like a sign of pain. And it's not. I mean, it's definitely not like a jab or anything for sure. They're not going through like heavy, like painful experiences, but it's kind of more of an annoyance. And so the faster they move, and the diameter of these little pricks change. So the bigger it is, of course, the more it's going to hurt. And so, when it was like a thinner one, that's when we saw, like, after 24 hours of kind of this withdrawal, the smaller ones, they were more sensitive to it compared to animals that didn't drink any. They just had water. And another cool thing, which I'm going to throw in this because this is something that I love, that one of the other grad students in our lab, who used to be in our lab, she's now going to become a professor in San Diego. She found that pain, the withdrawal of pain, actually is socially transferred. So if a mouse or a prairie vole is in a cage and another prairie vole or mouse are in the same room as them, that the other animal will actually show kind of a heightened pain sensitivity, even if they're not even drinking. So that's a whole other thing. That's a whole other
serious trend. Wow, that's amazing. I have just like a logistics question that might be. Did you wild capture these voles, or do you raise them in the lab?
Yeah. So
does that change anything?
So that's yeah. So that what we did, we raised them in the lab, but other labs across the U.S. actually go out and wild catch them. And so that's the cool thing about with prairie vole research is we try to make them as outbred as possible, because like mice and rats are usually inbred and they're always breeding with each other. Yeah. Voles, we try to. There's this kind of community. You kind of like take voles from other labs and try to make your genetic diversity different in your lab. So trying to make it, again, more kind of humanistic-esque. So you see so many different variabilities. So it makes it a lot harder because they're not the same, but it's like, this is what we want though, because this is what we see in humans.
Doing vole swapping.
Yeah.
You send them in the mail?
No, there's definitely like specific ways to send animals across. Oh yeah,
that's right. Like mammals and backbone animals have a different level other than fish and stuff like that. Cool. Well, I just want to put it out there for anyone who's listening to make sure if you have any other questions to please put them in the chat, either on YouTube or Facebook. Otherwise, I'm going to ask more of my silly questions. But one that I think I just wanted to know, as an educator of like young children, what or who inspired you to enter the STEM field? What pushed you to get involved? Yeah,
so what really got me involved was definitely in high school. I mean, I think I always knew I wanted to be kind of in science and STEM in general. So I went into college as the typical science person, being like, I'm going to be pre-med, I'm going to become a doctor. And then I realized that it wasn't for me. I was like, this is not what I want to do. I love working with people, but I don't think I wanted to work with people in kind of that way. And so I had my first lab experience in college that I was fortunate enough that someone was like, hey, are you interested in doing neuroscience? And I was like, sure. And I didn't know what a PhD was. My parents, one finished high school, the other one didn't finish high school. So I was just like, what's a PhD? How do you do this? can I do this? So my inspiration was kind of understanding just how I love understanding why people do things. And so that's why I became like a social neuroscientist. So yeah, my motivation was more that someone sparked an interest in me, and I was like, oh, this is cool. I want to know why things work.
Yeah. I had a little bit of a different directional situation. I definitely didn't think I was going to be a science person, and ended up studying science, but I also haven't gotten a PhD, so I'm not an ambitious science person. Way to go on sticking to it and like seeing it through. That's really awesome.
Thank you.
We had a question come in. Let me read this to you, and hopefully it will make sense. So studies on oxytocin in individuals with autism, positive results have been very hard to reproduce. Have you done your studies in totally different populations other than those that you raised in your labs?
No, we have not. And I think that is something that does need to happen, is having another lab do the same studies, or having, let's say, so these studies were done in 2017 and 2018. And so having, let's say, someone now, I think they have new animals now, or like 2 years from now, do these studies and see what happens.
Yeah. I've always been interested in the idea that science, like the proper experiments, should be repeatable, but I wonder how often they actually get repeated, because there's so much pressure to publish new information and something a little bit different, and to get the funding also in order to repeat an experiment. It seems like it's one of the many things missing in the science community, in the science process.
Yeah, definitely. I know, I agree. I think there needs to be reproducibility, because if you don't have things reproduced, then how do we know it's going to work?
Yeah. Or that it wasn't a fluke that it worked this one.
Yeah.
Awesome. How long do Prairie Voles live? How long could you raise your Prairie Voles for, and then it'd be a whole new generation or so?
Yeah. So Prairie Voles, they live, so in laboratories, they live for about, let's say, 2 to 2.5 years.
Okay.
Yeah. And then in the wild, of course, they're prey. So not very long in the wild for sure.
Okay. That's part of that evolutionary one after the other
Keep reproducing. Yeah.
Gotta go, or else you're going to turn into, I know we've dissected a lot of owl pellets, and most of the time it's a vole. I don't know why they keep getting eaten. Maybe there's a vole farm near the owl place. Another question came in that was, how long did the sensitivity to pain last after the discontinuation of alcohol?
Oh, good question. So my study I did mine only after 24 hours. But in mice, it's so funny. This is one of my best friends too that has ran the study. If I get this wrong, she's like, why do you know this? I think she did hers up to 4 days. And the other thing was not only she saw that social transfer of pain, but what she did was take kind of the bedding from one of the animals and put it into another room. And it's definitely olfactory that's causing the social transfer of pain. Wow.
And the alcohol doesn't, how long does the alcohol, do they metabolize the alcohol and pee it out, I guess?
They do it pretty quickly.
Okay. That's really interesting. I had another question that I could. Oh, you said that you give them 5% alcohol. Is that like a lager, or is it flat beer? Is it cider?
Yeah. So the way it's mixed is that's actually I've never got that question, and I don't understand why I never got that question, because it's a good question.
The mixed drink, grass mixed drink.
Yeah, it's I was a vole tender, and so it would be say a 100% ethanol and putting it into just straight tap water.
Okay.
And that was the mixture.
Okay. Nothing very exciting.
Yeah, nothing. wasn't adding some bitters or anything to it.
Okay. And I assume it's not colored then, so
Yeah.
Looks exactly the same. Yeah, you'd have to use vodka or something otherwise. Okay. Cool. Let's see. Anyone have any last burning questions, or else I'm going to ask one last question of Dr. Walcott. What do you think is like the most important, or there's so many different important things to do. What's one of the like best ways that educators or family members or community members can support marginalized students to enter the STEM field and feel confident and hopefully respected and get that voice at the table?
Yeah, I would say my biggest thing is it's definitely that representation, like seeing someone who looks like you in a field that you're aspiring to do. I mean, there's definitely not many scientists of color in Portland. So I try to give as many talks as I can as possible and try to accept everything like, hey, I'm here, like I want to talk and I want to try to put my voice out there and be a role model for people, and seeing that some people might tell you, no, you can't do this, but you really have to, like, you see someone and it's like, you can do this. Like, no matter what, you can do this. There's no specific, like, oh, this is what a scientist looks like. Scientists look like everyone. So yeah. I think that's the biggest thing, is getting that representation out there and make sure that individuals and kids see someone who looks like them in these spots, in these fields.
Thank you so much. It's been wonderful to meet you. I've heard a lot about you from your local fan club at OMSI. So I'm so glad that you came and talked about Prairie Voles. I had no idea about all of this stuff. It's really amazing, and there's a lot to learn, and I'm sure there's still lots to learn that other folks can keep on doing, as you suggested at the end of your talk. But I just want to give you a big round of applause and thank you so much for joining us today.
Thank you for having me.
Yeah. So I'm going to close out the end of our virtual science pub today by, again, thanking Dr. Walcott and just letting you know I hope you enjoyed tonight's event. If you'd like to watch the video again, or if you would like to share it with your friends, you can check out the video section on OMSI's Facebook page or YouTube channel. We have all of our old science pubs there as well. And don't forget to follow us on social media channels for updates on future events and inspiring content from OMSI. I know I mentioned it at the beginning, but please consider supporting Science Pub and making a donation. You can join us for the second to last virtual Science Pub in 3 weeks on Tuesday, July 6th, for a look at the rural-urban divide in Eastern Oregon through the lens of climate innovation. We'll hear from the past and present executive directors of Lake County Resources and watch a 29-minute award-winning documentary, Other Side of the Hill. Once again, thanks to our awesome partner, Celestream, for helping make tonight's event possible. We couldn't do it without you. And as always, you can get more information on our website at omsi.edu. Thanks and have a great night.